This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. From Microbe TV and the American Society for Microbiology, this is TWiM. This Week in Microbiology, episode 142, recorded on December 22nd, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today for this last episode of 2016 from Small Things Considered, Elio Schaechter.
Well, hello there.
Can you imagine another year has passed?
It's incredible. How many years have we had of this? About 6 years?
I think that's about right. Yeah.
Yeah, boy, it seems like yesterday.
It goes fast. That's right.
It does.
And also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello.
Welcome back. We missed you last time.
Yes, and there's no truth to the rumor that I didn't join because Michigan lost their football game.
No, we would never spread such a rumor. I'm so sorry. It was such a heartbreaking game, right?
Let's not talk about it.
Well, you have our condolences, but
thank you.
Life goes on.
It does.
And microbiology continues microbial sciences continues. And this is our last episode for 2016. It's also our last episode brought to you by CuriosityStream. So we move into a brave new world. Today we are Michael-less. Michael is visiting his siblings, which is what many people do over the holidays. And I like to stay home because it's too crazy to travel.
Yeah, especially up and down the East Coast, huh?
Yep. It's too actually. Although I think Thanksgiving is worse to travel than these holidays.
It's more compressed.
It's all compressed. But I have to say, the last little bit here, all three of our kids are home.
Oh, great!
For the next month.
Nice. It's
so different. When they're back versus not. And this is the first time they've all been gone. So
it's a lot more cooking.
It is a lot of cooking. But, we went to the movies the other night, this 3 kids and myself, and I realized there are not too many times remaining, and then we'll be able to all 3 do that just on a whim,
That's wonderful.
Anyway, we have today a snippet and a paper, and we'll start with a snippet brought to you by Elio.
Okay. Let me start out by saying the general point of this paper, which is called phenylpyroles, 30 years, 2 molecules, and in parentheses, nearly no resistance. And it's by Jafar Kilani, Sabrine Fillinger, both from the University of Paris in, Saclay. It appeared in Frontiers in Microbiology. Let me make a general point. We hear a lot about antibiotic resistance by bacteria and how we are facing a disaster. And I must say, I don't really know how much hype there is in that. It's obviously a big problem. Whether it is a potentially catastrophic problem, I don't really know. However, here is a glimmer of good news. It's just a glimmer. It turns out that there are drugs which are used against fungi in agriculture. They're sprayed on plants, on leaves, and so forth. They're called phenylpyroles. And they are used a lot. And guess what? With very few exceptions, you practically never find resistance to these drugs in fungi isolated in the environment. Interestingly, if you grow them in the lab, you have no problem whatever getting resistant mutants. So this is really a very interesting situation. What happens? Well, let me give you sort of the punchline. There is no real answer, but the possible punchline is that in order to become resistant, you have to invest a certain amount of energy in making the appropriate proteins, the appropriate enzymes, whatever, and that costs you. And it costs you in terms of what's called fitness. In other words, if you're a resistant bug, obviously in the presence of the drug, you're going to outgrow the wild type, of course, because the wild type is sensitive, the mutant is resistant. But in the absence of the drug, the wild type will win over because it is more fit. That is, it has a better chance of growing, and it will outgrow the mutant. Now, this has kind of been known in a sense, but I must say there is not many examples of this playing out in the environment. You want to comment on that, either of you?
Michele?
No, thank you.
Okay, then I'll go on. Okay, so let me tell you a little bit about this paper and about these phenylpyroles. Phenylpyroles are very simple molecules. They're a benzene ring with a benzene ring with a pyrrole attached to it. There's an original molecule called pyrrolonitrin, which is made by a secondary metabolite made by bacteria from tryptophan. It was isolated from a Pseudomonas in the 1960s, and it has strong antifungal activities against a whole collection of both animal and plant pathogens, even under greenhouse conditions. Now, this compound has been modified so that it contains it's modified by side groups being different. In one case, it's got fluorine instead of chlorine. These things are chlorinated compounds, by the way. Anyhow, so there are two compounds which have been used in the field. If you want the name, they're called, I'd never heard of them, phenpiclonyl and fludeoxonyl. So anyhow, they're used, and they are used widely. They are made by Ciba Geigy, now called Syngenta, and they are used for treating both seeds and leaves. How do they work? Well, that's kind of controversial. There's a lot of noise being made about it, and there's something definitive is not known. But it looks like they affect the polarity of the cell membrane of the fungi, so that they take in a lot more solutes, okay? And they essentially become loaded with it and loaded with water, and they burst. How this works is apparently through a protein kinase, which has a fancy name of hybrid histidine kinase, a typical fungal protein, which is involved in the proper balance of the cell membrane. With the drugs, this is affected in such a way that, as I say, the fungi die. Now, the mutants, as I told you, have largely been made under laboratory conditions, seem to be involved in this kinase, but that is not entirely clear. And for our argument, it doesn't really even matter. So essentially, that's the story. There is curious things that happened. The authors point out with reason that the strong fitness penalty of becoming resistant reduces sporulation, affects osmosensitivity, and makes the fungi less pathogenic. So so you become resistant to the drug, but you can't go anywhere. You lose your ability to survive in the environment versus the wild type. Another thing which is peculiar is that these compounds, which are effective against human pathogens, have not had medical applications, and the authors kind of wonder why. And in addition, the compounds that are in use have not been modified greatly. Normally, compounds which are effective in pharmacology get all kinds of modifications which make them more useful. Apparently, they talk about it as this was sort of a forgotten end of antifungal therapy. Anyhow, it's an interesting story, and I thought it is particularly interesting because, as I say, it's a ray of hope in the sense that resistant mutants have a harder time surviving in the environment. By the way, that's known for bacterial mutants as well, that they have a lower fitness. So it doesn't help the person who is sick or perhaps his immediate, or the plant, or the immediate plants or animals around it. But in the long run, it makes for what it says is that if this is true generally, and if we stop using a certain antibiotic, then the resistant mutants would disappear. Of course, when we use it again, it starts all over.
Right.
But that's, it's part of the equation. It's part of what one has to think about in terms of antibiotic resistance.
So the bottom line is that these are extensively used in crops, and we don't get resistance because the mutants that arise just can't compete out there, right?
Well, that may be the reason. Even that's not totally clear, but it's a possible reason. It's a likely reason, let's say.
So this target, this histidine kinase, is there anything like it in bacteria that one could explore as a target that might have a similar fitness cost?
Oh boy, that's a good question. I don't really know. I mean, bacteria have histidine kinases, plenty.
Yeah.
Whether they're analogous to these, I'm sorry, I don't know.
Well, that would be one approach to see if there's if there is an orthologous pathway, and then maybe try it. Right?
Although it's worth pointing out that although we hear a lot about antibacterial or antimicrobial resistance in bacteria, it's also a huge problem in fungal infections, and we have fewer drugs to treat fungi. And they cause some very challenging infections. So this is great news.
So, but you're thinking, Michele, to use this as a target in human fungal infections. Although as Elio said, why don't we use these drugs? Are they toxic, or have they just not been tried?
I don't know. I don't know. I haven't looked into it, but the authors talk about it as if it's weird that they haven't been used.
Because they're quite effective, clearly, and you would think that people had tried them. But there's no mention of it here.
Yeah, I wonder if they are toxic for humans.
They might be, but you could imagine that you could find non-toxic inhibitors. You know the target, so you could just do a screen for something that's not a problem. Well, anyone listening, working on this, here, read this and get some ideas. Really interesting. Yeah. Now, Elio, the bacteria that produce these compounds, the pyrrolnitrin, presumably, and if I'm wrong, tell me, presumably this is to inhibit competitors in the wild, fungal competitors?
I wish I knew. I'm sorry, I don't. I didn't look into the old history. Apparently, these compounds have been known since the 1960s.
Yeah.
There's a paper from 1965 that's quoted here, and I didn't look it up, so I don't know. I mean, but I suspect that nobody knows.
Yeah.
That the answer is not forthcoming.
Yeah, I always assume that these inhibitors, and of course, they're produced all throughout the microbial world, that they are there in part to deal with competitive aspects of growing in a complicated world. But who knows? Yeah. And it's really interesting. Well, that's a thought-provoking piece.
Can I make a totally relevant point?
Yeah, sure.
All of us peruse the tables of contents of journals, right?
Yes.
So we look at all kinds of things. And it struck me that a number of journals in microbiology have an increased number of articles on eukaryotic microbes. There's a lot more about protozoa and about fungi than I'm used to seeing. It used to be that it was all bacteria. And now there seems to be a lot of activity in the eukaryotic microbes, and it's probably a good thing.
I agree.
So we may hear more about it, is the point.
You think that's a real effect, or just a matter of the journals realizing?
Don't go by what I say. I mean, this is totally...
I think the technology, the mechanisms that we can use now to manipulate fungal pathogens have really advanced, and so there's more people doing kind of state-of-the-art mechanistic studies, would be my guess.
Good point.
Yeah, so I just did a TWiVo with a number of investigators at the University of Utah, and one of them works on Cryptococcus, and she said she worked in a lab where they developed the genetic system for Cryptococcus because there wasn't one, so you have to make the commitment to do that. But once you have it, then you can do a lot of things, which you couldn't do
And again,
before.
the disease burden is really quite high.
Yep.
And our choices of antifungals is much more narrow than antibacterials.
Yes.
Right.
Yeah, fungal infections are on the rise over the last decade, and it's not going to change, especially as more and more people are immunosuppressed. We got more and more fungal infections, and as Michele said, we don't have a good arsenal. So maybe this helps. Well, thank you, Elio. Quite provoking there.
Of course.
This episode is brought to you by CuriosityStream, the world's first ad-free nonfiction streaming service with over 1,500 titles and 600 hours of content. If you listened every hour of every day, it would take you 2 years or more to get through the whole library. It was originally founded by John Hendricks, who was from Discovery Channel, and that means you're going to get real science. It's not science fiction. It's real science, which is like we do here on TWiM. Real science, not fake science, okay? And that's CuriosityStream. How do you watch it? Well, you could go to a website on your computer or tablet or phone and just type in the URL, curiositystream.com, and play it there. But if you'd rather relax in front of your TV, especially this holiday season in front of a roaring fire, you could use your Apple TV or your Roku or your Chromecast device. There are a bunch of devices that allow you to watch internet programs on your TV. It's available in every country in the world. Well, Dixon de Pommier tells me 196 countries is all of it, but I'm waiting to be corrected there, as we always correct Dixon. And what they have is a wide variety of documentaries, science, technology, nature, history. They also have interviews and lectures. I'm thinking they should put my virology lectures on, don't you? Don't you think? Some of the features of their content: Stephen Hawking's favorite places. This is a relatively new documentary where Stephen pilots a computer-generated spaceship across the universe, and he stops at his favorite places. One of mine would be Tatooine or Jeddah. That would be cool to go to. Hint, that's a reference to Rogue One. Another one is Digits, a 3-part series hosted by the creator of Veritasium, which is a YouTube science channel. That's Derek Mueller. And it focuses on online safety and security. Yeah, that's a big one. And it features interviews with former NSA contractor Edward Snowden and co-founder of the internet, Vint Cerf. Deep Time History is all about the 14 billion year history of the universe. And we have underwater wonders of the national parks. You go under the bodies of water in these parks and figure out what's down there. Very cool. They also have super high definition, the so-called 4K. They have about 50 hours plus of 4K content. Now, how do you get this? Well, they have monthly and annual plans available. They start at just $2.99 a month, less than a cup of coffee every month. It's cheap for the content that you're getting. However, you don't have to pay right away. Listen to this. Check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiM. All right, now we have a paper which was originally sent in by our listener, Paul, who came across it and thought it might be interesting. And I agreed. And Michele has graciously agreed to talk about it with us.
And it's lovely. This is a paper published in November of 2016 in the Journal of Bacteriology, an ASM journal, titled The Single Transmembrane Segment of Minimal Sensor DESK Senses Temperature via a Membrane Thickness Caliper. And it's from the group, namely Maria Inda, Rafael Oliveira, Diego de Mendoza, and Larissa Cybulski. And they work in Argentina at the National Universities of Rosario and the National University of Cordoba. So, Cordoba. I know. I was going to ask you just to say all the names because you say it so much more beautifully, Elio. So a caliper is a device that's used to measure the distance between two opposite sides of an object. So in this case, we're thinking about how a bacteria can measure the thickness of its membrane. So as background, let me remind you about membranes. They are 2 layers, each built from phospholipids. So these are Jekyll and Hyde molecules that are composed of 2 long fatty acid chains that repel water, and they're joined to one head group that loves water. So the outer and inner surfaces of these bilayers are hydrophilic. They're stable in aqueous environments, such as our body fluids or the cytoplasm, whereas the interior of the membrane is fatty, and that's what holds it together. It's repelled by water, so that provides structural support. So inserted and sometimes even floating in these membranes are a wide variety of proteins that control the transport of not only materials across the lipid bilayer, but also the flow of information across the bilayer. So the class of membrane proteins that we'll talk about in this Journal of Bacteriology paper are messengers called 2-component regulatory systems. These are switches that are widespread in the microbial world. An E. coli cell, for example, has more than 50 of these 2-component regulatory switches. So 2-component systems have evolved to monitor a particular feature of the environment and then transmit its report to the cell's machinery. So it might sense temperature or osmolarity or a nutrient. In a typical 2-component system, there's a sensor protein embedded in the membrane that transfers information to a transcription factor in the cytoplasm. That transcription factor then turns on or off the appropriate response pathway, hence it's called a response regulator. So the language that these 2 components use is phosphate. In response to a particular environmental cue, the sensor that's embedded in the membrane will decorate itself with a phosphate. In other words, it's a kinase that can autophosphorylate, and then it will transfer this phosphate to the response regulator, whose ability to bind to DNA then increases so that it can either turn on or turn off the appropriate genetic circuit. So these 2-component systems, as I said, they're very widespread in the microbial world. They've been studied for decades. We understand in great molecular detail phosphorelays, DNA binding sites, the output or regulons of dozens of these 2-component systems in dozens of different microbial species. But only some of the environmental cues that actually trigger them are identified and understood, and the molecular details are much more murky. So that's where this paper has made just a beautiful advance. So Larisa Selbulski and her colleagues have deciphered an exquisite biochemical mechanism of how a 2-component system can equip Bacillus subtilis to adapt to cool temperatures by modifying the fluidity of its membrane. So when it's cooled, its fatty acids don't harden, like bacon drippings that we leave behind in the fry pan. So the main player is a membrane sensor, DESK. At low temperatures, it's a kinase. It will phosphorylate the transcription factor DESR, which in turn activates expression of a gene that encodes a fatty acid desaturase enzyme. So at warm temperatures, the DESK sensor can do the opposite. It can pull a phosphate off the transcription factor and turn off expression of the desaturase enzyme. So that means that the sensor is both a kinase and a phosphatase. And again, this is a very common feature of these 2-component systems, and it's one that this group exploits for their analysis. But how would altering the saturation of the membrane fatty acids help the bacterium? Why are they controlling this desaturase enzyme? So recall that fatty acids come in 2 flavors or 2 varieties. If they're saturated, all of their carbon bonds are single bonds. So butter has a lot of saturated fatty acids. That means it's solid at room temperature. It's got a higher melting point than, for example, olive oil, which has a lot of unsaturated fatty acids. That means it's got some double bonds in its carbon tail. Olive oil is rich in unsaturated fatty acids, so it's liquid at room temperature. It's got a lower melting point than butter. So these concepts of thickness of the fatty acid being dependent on temperature are something that we're going to return to. Now, the fatty acid desaturase that's controlled by this 2-component regulator is able to remove hydrogen atoms from a fatty acid, therefore shifting that fatty acid toward being more unsaturated. And in doing so, it can make the membrane more fluid at cooler temperatures. So the question that this group was investigating is, how does this protein that's sitting in the membrane equip bacillus to sense and respond to temperature by changing its membrane? And they had some key clues from previous work.
Let me interrupt for a second.
Sure.
We should point out that this protein, this particular sensor, is been a subject of a lot of study by this particular set of investigators. There's a whole literature, and I was surprised because I didn't know about it.
Yeah,
so
it's really a lovely body of work.
Yeah, there's a large body of work on this.
So the key clues that Elio points out from their earlier work are that they didn't think that DESK was sensing temperature per se, and the reason they were had to think about other environmental cues is that they could do an experiment and hold the temperature constant, but then change the fatty acid composition of the membrane, and in doing so, alter the sensor activity. So they could either activate or repress the sensor activity by simply changing the fatty acid composition, even when the temperature was held constant. So this led them to think about the fatty acid composition as what was actually being sensed or measured. They had also done some genetic studies to zero in on a couple of amino acids in the part of the sensor molecule that sits at the boundary of those Jekyll and Hyde phospholipids. So at the boundary of the hydrophobic tails and the hydrophilic heads. So imagine that DKSA has got a couple of amino acids that are sitting right up at that boundary. And in particular, I'm going to return later and talk about a lysine at position 10 and a phenylalanine at position 8. So with this as a foundation and other data that we won't go through, they built a really powerful tool. So instead of working with the full DESK protein, which loops through the membrane 5 times and would be quite complicated to purify and work with in a pure system, they built a simplified sensor. And this was comprised of just the first 17 amino acids of DESK that sit at the very top of this sensor near the interface of the hydrophilic and hydrophobic domains of the phospholipids. And then they fuse that directly to the last 14 amino acids of its final 5th membrane-spanning domain, which in turn was fused then to the catalytic cytoplasmic domain, or the business end, that either can phosphorylate or dephosphorylate the transcription factor. So with this simplified version, they were able to do a number of experiments that really advanced the field, because primarily a lot of our studies of 2-component systems use as a readout the transcriptional output. So imagine that the environmental condition changes, and then you can read through, say, a beta-gal fusion assay whether your target gene is turned on and off. But that is basically the sum of not only the sensing activity, but also the phosphorelay efficiency, the DNA binding, etc. So by using this minimal sensor, they could really zoom in on what is the response of this sensing domain that sits at the interface of the hydrophobic and hydrophilic domains of phospholipids, and how does that domain affect whether this protein is acting like a kinase or a phosphatase. So they performed a precise series of structure-function experiments, and here's the punchline. They were able to deduce that the DESK protein acts like a caliper that measures the thickness of the phospholipid membrane, a property that is temperature sensitive. And this is intuitive. Think of a little pool of olive oil spreading on a pan as you warm it up. The olive oil thins. So temperature affects the thickness or thinness of these fatty acids. So they were able to identify the particular amino acids and especially the side chains of these amino acids that equip DESK to measure the thickness of the membrane.
So, Michele, the thickness is measured in terms of the enzymatic activity of the protein, right? That's the output?
That's the output.
Yeah.
The thickness dictates whether it's in the kinase mode or the phosphatase mode.
We don't know how that works, right?
We don't, but they're going to give us some clues about how it works. Although how that signal is propagated to the enzymatic domain is still to be determined. All right. So as I mentioned, a key advance was developing this minimal sensor domain for DESK. They first needed to demonstrate that it retains the capacity to increases or decreases in temperature by activating or repressing its target promoter. So they first did this by expressing that minimal domain in live Bacillus subtilis, and then they measured the output using classical fusions of the desaturase enzyme promoter to beta-galactosidase, and that's shown in Figure 1. The next step they wanted to test before they got into their structure-function studies was to ask, does the thickness of the Bacillus subtilis membrane actually change in thickness in the physiological temperature ranges from 20 to 40 degrees? So for this, which they show in Figure 2, they employ a biophysical technique called small angle X-ray scattering, which basically allows you to capture information about the structure of molecules or materials without having to crystallize them or fix them. So they do this by shining the laser at a very low angle toward the material and then capturing the reflection and then doing a lot of computation to translate that into differences in shape of the protein. So they were of the lipids. So they were able to detect a subtle change of just a single angstrom in the thickness of these Bacillus subtilis membranes between the temperatures of 25 and 37 degrees.
I think this is phenomenal.
It's amazing, isn't it?
Nice.
To put that in context, 1 angstrom is 10 to the minus 8 centimeter. But let's think in biological terms. 1 turn of a DNA helix is 34 angstroms. So now we're looking at a fraction of a DNA helix. And then for this paper, back to Vincent's point, thinking about the protein structure is even more relevant. So an alpha helical
Can I make a point
protein,
here? When I look at the graph,
please.
What's kind of amazing is that the changes are very small.
Very small.
However, they're very accurately, you can measure them very, very accurately. So this is a fantastic technique because it allows you to measure with precision rather small changes.
It's really incredible. And again, to emphasize how small these changes are, if we think about an alpha helical protein, each amino acid rises in the helix 1.5 angstroms. So that is how sensitive this method is, and they're getting about that 1 amino acid shift in the membrane thickness equivalent.
Had you ever heard of this method, either of you? I hadn't.
No. As I tried to think about it, I was reminded of like looking for something I've dropped on the floor, like maybe a contact lens or a pin. And when I'm standing up looking straight down, it's hard to see it. But if I put my head right down on the floor, sometimes I can detect the bump in the floor. So I think that's the small angle X-ray scattering. That's the principle, I think. Anyway, the instrument's very sensitive, and they did detect this reproducible, tiny increase in the thickness when the membranes were cooled. So that motivated them to go on and test whether this sensor protein could actually detect that 1 angstrom difference.
I have to say, Michele, this graph, they have the y-axis in angstroms. I've never seen.
Yeah.
Can you imagine? Because
that's a small number.
Because of 29.5 to 28 angstroms. Oh my gosh. It's incredible. Yeah.
It's the inset. Yeah.
Yeah, that little inset. Yeah.
And the points fall on the line like you would. Oh. It's a dream. Yeah. This is a dream result with a technique that none of us ever heard before.
It's true.
Amazing.
So now the question is, can a bacterial protein actually respond to this subtle change in membrane thickness by turning a gene on or off? So to ask that question, they prepared membrane vesicles with their minimal sensor DESK embedded in them. So this too was a real technical advance. The way they did it was they had their phospholipids, they solubilized them in detergent, then they added their purified minimal sensor protein to the slurry, mixed those together, and then they added beads called biobeads that could bind the detergent, remove it, and in doing so would force the phospholipids to then form little vesicles or liposomes that had incorporated now their minimal sensor DESK protein. So they verified that, in fact, the sensor protein was embedded in the membrane and not simply stuck to it by bathing their liposomes in high salt, which would disrupt electrostatic binding, it was still associated. They then did some amazing experiments. They verified that their minimal sensor DESK construct retained both its autokinase activity and its phosphatase activity, and it still responded to shifts in temperature from 25 up to 37 degrees. So they had a really beautiful in vitro system with which they could now do structure function analysis, begin to change amino acids, and ask what properties of this amino terminal region was conferring this hypothetical caliper activity. So in particular, they focused on an amino acid lysine at position 10 and then a phenylalanine at position 8. And they altered them to other amino acids that had similar hydrophobicity, similar charge, etc., and then tested the activity of the sensor domain, either using their kinase or phosphatase activities, or in live cell assays, or in the liposome model. So they had a lot of assays available to test this model. So for example, when they replaced the lysine at position 10 with leucine, they saw that its kinase activity decreased and its phosphatase activity increased. If they added an extra lysine at position 11, so right next door, they got an increase in increase in kinase activity and a decrease in phosphatase activity. So they're beginning to feel like they had some insight to what properties of this region of the sensor was key. But how is this working? So we have to think back to these Jekyll and Hyde phospholipid molecules. The head of the molecule loves water. The tail hates water. So what they did was investigate whether the side chain of the lysine at position 10 had what are called in the field snorkeling properties. This is a term I hadn't heard, but I love it. So a snorkeling property, snorkel, like we snorkel for looking at beautiful fish in warm waters. And it refers to the extension of the snorkel up into the air from the water. So in this case, think about that lysine sticking its side chain out of the lipid and up into the hydrophilic part of the molecule. That's what they've deduced. So if they change that lysine to a glutamate, so it's still hydrophilic, but it's negatively charged, they saw less kinase activity. If they changed it to an arginine, so it retained its hydrophilic character and it retained its positive charge and a side chain that too could poke up into the hydrophilic region, they got no phenotype. So this gave them, they deduced then that the capacity of this amino acid at position 10 for its side chain to snorkel, to stick up into a hydrophilic environment, they postulated that it was pulling the rest of the sensor protein with it, inching it up higher, not inching it, budging it up into the away from the cytoplasm and up toward the extracellular region. So that was one residue that defined this caliper. The other residue is the phenylalanine at position 8. So this is a hydrophobic residue, and they deduce that it acts like a greasy cap that locks the DESK into the fatty acid region of the phospholipid membrane. And they concluded that because if they converted the phenylalanine to alanine, which is less hydrophobic, the molecule lost its ability to sense. It couldn't phosphorylate at either 25 or 37 degrees. If they change it to a leucine, which is a little bit more hydrophobic, it now had some kinase activity and it could respond to temperature. And then finally, if they changed it to a tryptophan, which is a more hydrophobic molecule, they found it had kinase activity at both temperatures, both 25 and 37. So in essence, they found that the hydrophobicity of the molecule or the amino acid at position 8 was dictating whether or not the kinase activity was present and whether it could respond to temperature. So their model then is that at high temperatures, the membrane is thin and it's hydrated. And the side chain of that lysine at position 10 snorkels out to the aqueous phase. Meanwhile, 2 amino acids away, the phenylalanine is stuck in the fatty core of the membrane. In this configuration, the whole transmembrane segment that extends down toward the cytoplasm and the business end of the molecule is in a relaxed conformation, and therefore there is phosphatase activity by the sensor. When temperatures cool, the membrane thickens, and now that side chain of the lysine at position 10 is pulled into a more hydrophobic environment, but it can still snorkel out and reach the hydrophilic aqueous phase, and by doing so, it pulls the transmembrane helix outward. And they think that that pulling on the transmembrane region, due strictly to the love of the side chain for the hydrophilic region of phospholipids, is sufficient to propagate a force through that transmembrane spanning domain to the cytoplasmic domain and somehow shift the architecture to favor kinase activity. So that is the story, and it is built through really beautiful series of figures that take advantage of not only whole cells, but this liposome model to go in and test the impact of particular site-directed mutants. And because they have used a minimal system, they can ascribe the activities to that short just 17 amino acid sensing region, and in particular, the side chains on the lysine 10 and the phenylalanine 8. So I thought this was a really beautiful biochemical and genetic investigation that reveals a plausible mechanism for how Bacillus subtilis is able to respond to slightly cooler temperatures by altering expression of an enzyme that could change the saturation of its phospholipids and therefore keep the membrane fluid despite the lower temperatures.
Michele, what would happen if, and I'm sure this has been done, you introduce mutations that prevent this sensing of temperature? So at a low temperature, what would happen to the bacterium? Would it die or?
I believe that is true. I don't know whether it's bacteriostatic or lethal, but I would imagine a number of the other proteins that are embedded in the membrane that rely on fluidity, their activity would also be hampered.
So this was done with just one of those transmembrane domains.
That's right.
So what do they think the others are doing?
So they do, to their credit, in the discussion, they do admit that the situation could get more complicated when they add back the full protein. Nevertheless, what's an interesting implication of this work is that if you want to specifically design sensors for application, you can do it with just this minimal domain. So they're moving now not away from what is the biology of Bacillus subtilis, but how can we take this information and design biosensors that we could incorporate into switches.
I think the scale of this really helps us understand that at this level, at the molecular level, very, very small changes make a big difference. I mean, really, this is on the angstrom scale.
It does help to remember that the proteins themselves are on the angstrom scale.
Yeah.
They're also quite tiny.
Yeah, a lot of people don't remember that. And they wonder, how do these things happen? And this is a great illustration of that because you can see the readout of a 1 angstrom change. It's just cool. It's so cool.
And the paper was written beautifully so that they really walked you through the chemistry of each of the side chains and reminded you about their hydrophobicity and helped you imagine or picture in your mind how the molecules were shifting under the different conditions. So I found it very accessible. Listeners
may find the details a little bit intricate, but I would urge people to look at this paper because it is, in fact, it reduces things to a degree of simplicity that's totally unexpected, I think. So it has this patina of a lot of data, but behind it is a series of amazing, sort of straightforward, simple thoughts that are new, they're novel to me. So this is quite a paper.
It is. And I should give them credit. Each of their figures has kinetic data. So it's not just yes, no for this kinase activity or phosphatase, but they carefully do a time series and can therefore add sensitivity to their measurements and then really make strong conclusions about the impact of one side chain versus another at particular amino acid positions.
Wonderful. It's really nice. So
tell us about the first author.
Yes. Well, Michele did double duty here and got some information from Maria, who belongs to an Italian family that went to Argentina during World War I. You know, a lot of families were.
A lot of people did. That's not uncommon.
Yep. She says looking for peace. So they settled in Argentina, and her family were farmers for a while, and then her parents were able to go into the city and get university education and eventually become professionals. Then they settled in the city where Maria was born. And she says that perhaps because of this history, hard work has been a well-rooted value in the family. I have to say, my father came over from Italy to the U.S., and I think that was true of him as well. He had an agrarian background as well in Italy. She grew up liking school and did very well in school. However, she says she grew up in the shadow of her overachieving older brother. So whenever she did something really well, it wasn't quite recognized. She could finish first, but no one was surprised by that. Anyway, she grew up having a liking for education, and that was encouraged by her family. She says, we would ask your father why the trees are green. He would say, because of the chlorophyll. And the questions would go on and on. And she says, I've always preferred children to adults in their perception of the world. Maybe becoming a scientist is my way to avoid social condemnation towards my child's view of life.
Always asking questions, huh?
Yep, that's great. Well, I'm often told by my family to stop asking so many questions. I always ask questions.
I get that too.
I just think, how else do you have a conversation, right?
Right.
You have a conversation about it. Anyway, she went to school and was interested between computer science and biological sciences, but eventually the biology won out. She entered a biology Olympic competition and eventually the national and international versions and did very well. And she eventually appreciated that genetics was a great way to understand the beauty of biological systems, and that's why she decided to study genetics, as you can see from
it certainly comes through in this paper, doesn't it?
Yeah. So outside of her work, she likes to do a lot of things. She likes to draw, paint, take artistic photos. She plays guitar, sings in a chorus. She's involved in kids activity, and she practices aerial dance. My gosh, I'm getting exhausted. She said art helps her to develop her creativity. And in fact, she entered the ASM Auger plate art competition, which we mentioned here multiple times. I think was that last year or earlier this year? And they picked some winners, and I believe she was one of them. She produced a plate called Harvest Season, which she painted with colorful yeast mutants that depicted the development of a yeast field in a Van Gogh style.
Wow.
And this picture has appeared in many scientific and technological magazines with the idea, of course, of raising public awareness for science. Those Auger art competitions, in fact, have done amazing things. People are really getting engaged. And in fact, if you follow ASM's Instagram account, every day now they're putting a picture of an Auger plate because it's the holidays. They had one just today, which is a movie of the culture growing and forming a Christmas tree.
Wow.
It's really cool.
I have the Auger art calendar hanging in my kitchen, and I just love it.
It's very pretty, yeah. Frankly, I have to say, someone made an Auger art of my likeness.
Right.
And I think that should have won, of course, but it didn't. It was just such a good likeness. All right, moving along Maria's career, while she was getting her PhD, she was captivated by genetics and the power of molecular biology, but she still liked computer science, and she took a introductory course in synthetic biology. This is a EMBO course. And then she took some courses at Cold Spring Harbor and was able to present her findings at an EMBL symposium, enabling technologies for eukaryotic synthetic biology in Heidelberg. And she got an award for that. So now this has turned her on to the whole field of synthetic biology. That is where you can design organisms. We do that in virology. We can alter viruses, and you can also do it in eukaryotic cells. And so now that she's done in Larissa's lab, she's going to go to MIT to Dr. Liu and work on synthetic biology. She thinks it's a really important and growing field that's going to contribute a lot to our future understanding and our future world, things like biocomputing, biosensing, bioparts, biocircuits. So she's very excited to get into that field.
Got ideas about developing biocircuits that can be used for diagnosis and treatment of different illnesses.
Yep, absolutely.
That's cool.
She also has a bit of advice for junior colleagues. She has a proverb that she got from a friend of hers. Buy the truth and do not sell it. Consider truth as a thing of the highest value. Spare no pains, cost, or sacrifice to obtain it. And when gotten, keep it safe. Do not barter it for profit, as no one can be established through wickedness. Sell it not. As San Martin, our national hero, once said, you will be what you should be or else will not be. It's just great. And finally, she has a strong memory of interesting days in the laboratory. And she said, as you heard today, she worked her PhD thesis and postdoc to understand how the cold thermosensor protein DES-K is activated. And every time they thought about the results and how to interpret them, they always ended up simplifying the mechanism to a minimalistic version, which you heard. They made a single transmembrane domain. And she said they were always surprised by our results. Most of the time we thought we understood it, but then we found we were mistaken. And she says, I think that's the beauty of nature. So simple, so versatile, and so much perfection. So clearly a really thoughtful
nice
scientist.
Way with words.
Wonderful.
Yes, she does.
Really thoughtful,
Beautiful.
and I like that very much. So thank you, Michele, for reaching out to her.
Well, thank you, Maria, for providing that lovely bio.
Yep.
And
Really
beautiful
lovely.
biology that will really be influential in the field.
Yep. Beautiful work, and good luck in your next stage, Maria. I have a few interesting emails to wrap up this TWiM. One is from Carol, who writes, Dear Vincent and the hosts of TWiM, I am a longtime listener and fan of your weekly TWiM podcasts. I really enjoyed the latest episode in which you discussed a paper by Kelly Wrighton and colleagues, and was especially happy to hear you talk about chemistry. On that note, I'm writing to tell you and your listeners about a seminar series entitled The Chemistry of Microbiomes, organized by the Chemical Sciences Roundtable of the National Academy of Sciences. In separate workshops, the series addressed earth, marine, and human microbiomes, and we were fortunate to have Kelly Wrighton, one of the co-authors of the paper we discussed, speak at the Earth Microbiome Species. All these talks have been archived, and I'll give you a link for that. And there was a recent seminar on December 7th, which of course is now over, but there is an archive of all those talks as well. So you might want to check that out, because if there are future talks, you can always listen in and email or tweet questions during the talks. Wishing all of you happy holidays, Carol. Thank you, Carol. The next one is addressed to Michele. It's Mark writes, I am that rare thing, a British baseball fan, an ASM member, and I've been rooting for the Cubbies this last postseason. On top of their amazing win, my American postdoc, Morgan Feeney, just pointed out that in the very early 1900s, before they were called the Cubs, the Chicago National League team were briefly called the Chicago Microbes. Amazing. Due most likely to Chicago's famously bad sewage system of 120 years ago, basically the river. So he sends a link to an article in a website that I cannot mention on this show because we are a family website. But this is all about this, and they have headlines where they say, microbes alert with second place at stake. Microbes had a hot time. Oh, this is great. What's another?
I'm really sorry that Michael's not here, since he's the Chicagoan Cub fan. Yeah.
Microbes easy for the Cardinals. So they actually called this team the Microbes. How about that? That is
so we've won
great.
a World Series this year. The Microbes have.
Yep. That's from Mark, who's a professor and head of the Department of Molecular Microbiology at the John Innes Center in Norwich, UK. Wonderful. And finally, the last one, Elio, this is for you. Soupty writes, hello, I had a question regarding specialized transduction. What are the consequences of the portion of the genome of phage which remains attached to the bacterial chromosome as a result of faulty excision? Does it lead to any kind of useful mutation?
Gee. I got to think about this. I can't think of one. I don't know. I really have to think about this. Sorry.
Can you explain what specialized transduction is?
Well, specialized transduction refers to the fact that a segment of a lysogenic phage is incorporated into the genome of the host and carries along genes from a previous bacterial host.
Right.
So that's typically genes which are adjacent to the prophage and which are carried along with it. Mm-hmm. So it refers to, as opposed to generalized transduction, which is any gene is picked up by a phage, and any bacterial gene is picked up by a phage and can be transferred to another bacterium. Here we're talking about lysogeny, actually. So the question, to answer the question, is what good is lysogeny? And that's a complicated question.
Yeah, I don't know, because I just haven't ever encountered it. But the idea would be that, is there a function for the genome that's in the bacterial genome, basically, right?
Well,
he says it's a part of the phage, that genome that remains attached, right? Because it's faulty excision.
So if the excision is not precise, then some small bit will be left behind.
Yeah.
And so that might, in some cases, act like an insertion mutation, depending on where it
I guess it could, yeah.
Well, it's already being inserted. I mean, the gene is being carried, is already inserted. So I don't know that would happen.
Or you could change a function?
You could imagine that if it's in frame, it might change. Yeah, it could change. But I don't know of any example of it, right? That's the key.
I don't know.
Maybe if someone knows, tell us. That's a great question, though.
Yeah.
I like it. All right, that will do it for TWiM 142. This is the last TWiM for 2016, but we'll be back next year, hopefully every other week, and bring you more on the microbial sciences. You can find it, by the way, at iTunes, microbe.tv/TWiM, and also at asm.org, the ASM website. And consider becoming a patron of the show. You can go to microbe.tv/contribute. There are a number of ways you can help us out. You can use a Patreon mechanism and subscribe, or you can donate in a variety of other ways to keep our shows moving, to get some assistance for us, and to help us travel a little bit. We love getting your questions and comments. You can send them to TWiM, TWiM, at microbe.tv. Elio Schaechter is at the wonderful blog Small Things Considered. Thank you, Elio.
Well, thank you, and I wish everybody happy holidays.
Thank you. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you. Pleasure to be here.
And I am Vincent Racaniello. You can find me at virology.ws. I also want to thank Michael Schmidt, the Medical University of South Carolina, who was with us for much of 2016, for his participation. We miss you, Michael. We'll see you next time. I also want to thank ASM, the American Society for Microbiology, for their support of TWiM, Chris Condian and Ray Ortega for their technical help. Also want to thank the sponsor of this episode, CuriosityStream. The music you hear on the show, if you listen at the beginning and end, there's a little bit of happy music. That's composed and performed by Ronald Jenkins. You can check out the rest of his work at ronaldjenkins.com. I want to thank everyone who listens for listening in the past year, and I hope you have a great and happy holiday and a happy new year as well. We'll see you next year. Thanks for listening, everyone.
Yeah.
Want to wish everyone a happy new year.
Happy New Year.
Thanks for listening, everyone.
Happy New Year.
Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.